Hello everyone and welcome back to more human physiology. So for the rest of these videos for this chapter, we're going to cover just some basic cell biology, a lot of which you're going to need as you continue your study of human physiology. So let's get into this. So the first thing that, or one of the first things that you probably learned when you started talking about cells in general biology is that there are two major types of cells. You've got eukaryotic cells and you've got prokaryotic cells. Prokaryotic cells are very simple cells that don't really show a whole lot of internal organization. Usually you mean to say that the DNA is not contained within a membrane-bound nucleus. Well, if we go beyond that, you also don't find organelles like mitochondria, Golgi, endoplasmic reticulum, and the like. So, the main thing for us to understand, we're not really going to be talking about bacteria or any other kind of prokaryotic cell throughout the rest of the semester. Since this is human physiology, we are going to focus on human cells, and human cells are of the eukaryotic variety. And as such, we're going to need to be aware of the different organelles and what they tend to do. So usually each organelle is going to have some function that is going to overlap with the function of another organelle because those organelles tend to communicate with each other, or if they don't communicate with each other, they in some way coordinate their activities to accomplish a larger goal. Usually for most of these, you're talking about these organelles working together to synthesize a protein, which we will talk about a little bit later. So what I like to do, we're not going to talk about every single organelle in this video because some of that falls under the on your own umbrella that you're going to do on your own. But what I like to do is for each organelle, I like to use analogies that frame each organelle as kind of part of a city as a whole. If the cell is a city, you could say the nucleus is like town hall, mitochondria are like little power plants, the Golgi apparatus is like the post office, so we'll be using those sorts of analogies. So there's lots of different organelles for us to cover, so what we like to do is to split them up into three different categories. Number one, you've got the endomembrane system. This contains a lot of organelles. It's going to include the nucleus, the endoplasmic reticulum, the Golgi apparatus, and the ribosomes, and most of what you're going to see here is going to involve protein synthesis and a little bit of lipid synthesis. The lipid synthesis aspect will become important later on when we talk about how endocrine cells in the body synthesize and secrete steroid hormones, which of course are lipids. You've got your energy-producing organelles, which for us as humans, that pretty much is just the mitochondria. We are not plants, so we do not have chloroplasts, so that should be a pretty quick and easy discussion there. And then you've also got the cytoskeleton. The cytoskeleton does not actually comprise any membrane-bound organelles, but rather these are protein filaments that serve a variety of different functions inside the cell. The endomembrane system aspect here is mostly going to be on your own work, so most of our focus in this video is going to focus on the mitochondria. Now, just to give you a brief introduction to the organelles of the endomembrane system, so you'll have a little bit better idea of what you're dealing with in your on your own work. You've got the nucleus. This, of course, is the site of both DNA and chromosome storage. This is where you will see DNA replication occur just prior to mitosis. RNA synthesis, so you're also going to see the synthesis of all of our important RNA molecules like messenger RNA, which happens during the transcription process, and then also ribosomal and transfer RNAs. And I mentioned the transcription process of making a messenger RNA molecule from a DNA template. That is the transcription process, and that is the very beginning of protein synthesis, which of course happens in the nucleus. The endoplasmic reticulum is actually continuous with the nuclear envelope, so they're not actually two separate things, but the ER is actually just a continuum of the nuclear envelope, which is the nucleus. And this is going to be the site of membrane protein synthesis for ribosomes that are embedded in the rough ER, and then lipid and steroid synthesis if you're talking about the smooth ER. So it's good to understand that there is a dichotomy of functions there. Ribosomes are not actually membrane-bound structures. They're actually a conglomeration of ribosomal RNA and protein, and they work together to synthesize proteins from the messenger RNA transcripts that were put together in the nucleus. The ribosomes can either exist free in the cytoplasm, and that's where you will see a lot of soluble proteins being made. And the ribosomes in the rough ER are mainly going to focus on proteins that are embedded in the membrane. The Golgi apparatus is going to serve as a site of protein and lipid modification, and the main function of adding carbohydrate chains to newly synthesized proteins in this case is more or less to determine where those cargo are meant to go. Is the protein supposed to stay inside the cell? Is it supposed to go to the mitochondria? Are we supposed to get it to leave the cell by exocytosis? The Golgi apparatus helps to determine these things, which is why I like to think of the Golgi apparatus as being the cell's post office. And then the lysosome is going to serve as a major site of macromolecule degradation. Whether it's proteins or nucleic acids or carbohydrates, if the cell decides that a certain molecule is no longer needed, the lysosome contains a lot of hydrolytic enzymes. Those are enzymes that catalyze the hydrolysis or breakage of the covalent bonds that join these monomers together to make proteins, etc. So if we need to destroy a particular macromolecule, we can send them to the lysosome to do so. Okay, so let's get to the major focus of this video, which of course is the mitochondria. Now, the organelles that relate to energy production or general detoxification in some way are the chloroplasts, the mitochondria, and the peroxisomes. I've already said why we're not going to talk about the chloroplasts, because of course as humans our cells do not have chloroplasts and we are therefore not capable of photosynthesis. And we're not really going to talk about the peroxisomes. We'll talk about the peroxisomes a little bit later on in the semester when we get to the respiratory system. The peroxisomes basically work to detoxify a lot of oxygen radicals inside the cell. So since we're going to talk about the purpose and function of oxygen later on in the semester, we'll save that conversation for then. So really what we're going to focus on here are the mitochondria. Mitochondria, of course, are double-membraned organelles, meaning that they have both an outer membrane and an inner membrane with a small space in between. In this case, that space is called the intermembrane space. And mitochondria, of course, are the site of cellular respiration activities. So I mentioned that we've got both the outer and the inner membrane. You've got an inner membrane space, and then inside the inner membrane you have the matrix. So this is a special type of fluid that is going to contain all of the enzymes that are necessary for catalyzing the variety of different reactions that make up the cellular respiration process. It's important to understand cellular respiration is not one reaction. It is actually dozens of reactions, both glycolysis, which happens outside the mitochondria, and then all of the different reactions that occur inside the mitochondria, both the Krebs cycle and the electron transport chain. So cellular respiration is not just one chemical reaction. It is many, many, many chemical reactions that you have to get from A to Z in order to get your ATP. Now, if you've ever looked at cell diagrams that show you maybe one or two or even three of these little sausage-shaped mitochondria, just know that those diagrams are not exactly drawn to scale. So most cells in the body do have mitochondria, but most cells tend to have hundreds or even thousands of mitochondria instead of just maybe one or two or three. Now, cells can make new mitochondria, or they can destroy existing mitochondria by sending those organelles to the lysosome. How many mitochondria a given cell maintains is just going to depend on how much ATP that particular cell needs to make. Okay, so let's focus on cellular respiration, which we understand is going to heavily involve the mitochondria. The actual process of making ATP through cellular respiration is generally going to follow 3 steps, and you'll probably remember a lot of these from general biology. Number 1 is glycolysis. Glycolysis technically is not a part of cellular respiration because glycolysis can occur regardless of whether oxygen is present or not. This takes place in the cytoplasm, so it does not take place in the mitochondria, it takes place in the cytoplasm. And like I said, the mitochondria are actually not necessary here because glycolysis should always occur regardless of whether oxygen is present or not. And keep in mind, cellular respiration can only occur if oxygen is present. If oxygen is present, then the product of glycolysis, which is a molecule called pyruvate, will be sent into the mitochondria where those molecules of pyruvate will participate in the Krebs cycle. The enzymes that catalyze the different reactions of the Krebs cycle are located in the mitochondrial matrix, hence the reason why you've got to have mitochondria in order to proceed on to the Krebs cycle. So the result of the Krebs cycle is that we do make a very small amount of ATP equivalents, but the main thing that we're going to produce here are high energy molecules NADH and FADH2 for every turn of the Krebs cycle, and the Krebs cycle will turn 2 times for every molecule of glucose that you started with in glycolysis. For every turn of the Krebs cycle, you are going to make 3 molecules of NADH and 1 molecule of FADH2. Like I said, this will require the presence of oxygen. Oxygen is not actually directly used during the Krebs cycle, but the presence of oxygen has to be confirmed in order for the Krebs cycle to proceed. So, a major end product of the different reactions of the Krebs cycle is that we do make some carbon dioxide waste products. We've talked about this before. Carbon dioxide gets produced from cellular respiration. If that carbon dioxide builds up in the blood, it can change the pH of the blood, so we need to make sure that we are using our respiratory system to expel a lot of that excess CO2 so that we can maintain pH balance in the body. And then finally, the electron transport chain is going to take high energy electrons from those NADH and FADH2 molecules, and those high energy electrons are utilized to pump hydrogen ions that are found in the matrix of the mitochondria across the inner membrane and into the intermembrane space. This is a perfect example of active transport. So this is active transport because these hydrogen ions are going from an area of low concentration in the matrix to an area of high concentration in the intermembrane space. So that is why we are using the energy from NADH and FADH2 to push those hydrogen ions against their concentration gradient. So what we do here by doing that is we maintain a very significant hydrogen ion gradient. Hydrogen wants to move from high to low concentration from the intermembrane space back into the matrix, and when we allow hydrogen ion to move like this, it moves through a major complex called ATP synthase, and the energy that is released as hydrogen moves down its concentration gradient is used to synthesize new molecules of ATP. The electrons that are transferred from high energy in NADH and FADH2 to progressively lower and lower and lower energy by the different complexes of the electron transport chain, those electrons are eventually transferred to oxygen, which acts as the final electron acceptor here. We have to have oxygen to play this role, otherwise none of this is going to work right. So that explains why we can only make all of this ATP in step 3 here if the presence of oxygen is actually something we can confirm. This diagram here gives you kind of an idea of what is happening during the electron transport chain. NADH and FADH2 produced during the Krebs cycle are able to interact with different protein complexes that are found within the inner membrane of the mitochondria, and these complexes are able to perform redox reactions because these complexes tend to contain highly reactive iron. And as the electrons are transferred, we use that energy to pump hydrogen ions into the inner membrane space, until eventually those hydrogen ions go down their concentration gradient through the ATP synthase complex, and we use that momentum to make lots and lots of ATP. And you can see here, Oxygen acts as the final electron acceptor after these electrons from NADH and FADH2 pass from complex 1 to complex 2 to CoQ to complex 3 to cytochrome C to complex 4. Those electrons are now at a very low energy, and they are transferred to oxygen, which is now reduced to form a molecule of water. Okay, so let us do some bookkeeping here. So let us make sure that we understand exactly why having oxygen is so important, especially if you're talking about one of those tissues that we talked about in Chapter 1 that need oxygen a lot. Brain tissue, heart tissue, skeletal muscle tissue. Those are highly, highly, highly demanding tissues. They need lots of oxygen so they can make as much ATP as possible to fuel all of their activities. So, not to belabor the point here, you can kind of figure some of this out on your own. When oxygen is not available, we cannot move forward with the Krebs cycle, which is called the citric acid cycle here, and we cannot therefore go through with the electron transport chain. So what this means is that if oxygen is not available, the only thing we can do is glycolysis. And in glycolysis, you only make 2 ATP molecules for every glucose molecule that is consumed. Now, if oxygen is available, not only are you going to make those 2 ATP during glycolysis, you're also going to make 2 more ATP during the citric acid cycle or the Krebs cycle, and then you're also going to make all of this ATP during the electron transport chain. If you add all of those ATP molecules together, you're going to get about 36 molecules of ATP for every glucose molecule consumed. So the bottom line here is that you are going to make about 18 times more ATP if your cells have oxygen versus if they do not. And obviously, if you're talking about tissue in your body that needs oxygen a lot, like the brain and like the heart, you're going to want to make sure that you keep those tissues well perfused with oxygen so they can make as much ATP as they need. All right, that's going to do it for this video. I hope you found it useful. Join me next time for the last video of this series of Chapter 3. We'll talk a little bit about the protein synthesis process, but most of what we're going to focus on is how proteins can be made incorrectly in genetic diseases and how those mutated proteins can cause problems in physiological processes. Thanks again, and I will see you